Hello and welcome to Kulkarni Laboratory. My name is Mrs. Amruta Kulkarni and today I am going to give you a small presentation on protein content from food sample. First of all, what are proteins? So, proteins are very large molecules composed of basic units called amino acids. Proteins contain carbon, hydrogen, oxygen, nitrogen and sulfur. In short, we can say that proteins are nothing but the long chain of amino acids. Then, what are the sources of proteins? As you can see from this picture, there are meat products, cereal and pulses, fruits, almonds, etc. All these are rich sources of proteins. Further, I would like to mention here the proteins are divided into two categories like first class proteins and second class proteins. So now what are the first class proteins? These are nothing but the proteins which derived from animal products such as chicken, meat products, cheese, butter, etc. All these comes under first class proteins. And now the second one is the second class proteins. These are nothing but the proteins derived from plant products such as cereals and pulses, vegetables, fruits, etc. Now, how to estimate the protein content from food samples? The total protein content in almost all food and agricultural products is determined by Geldal method, the very famous method. Now, what is the principle behind this method? So whatever the reagents which we are using while doing the analysis by Geldal's method, that is the very much important is sulfuric acid. So in presence of sulfuric acid and catalyst, the nitrogen atom in the nitrogenous organic compound, that is your food sample, whatever it may be, is converted into ammonium sulfate. So the ammonia is then distilled from alkaline medium and it is absorbed in boric acid. This is the state which takes place in the distillation state that we will cover in the next slide. The ammonia is then determined by titration with standard mineral acid. So in short, you can see here the chemical reaction which occurs during this stages while determining the protein content. We'll see the briefly how the reaction takes place considering the protein as an example. If your sample contains proteinaceous nitrogen, so in presence of H2SO4 that is sulfuric acid, it will convert into NH4 twice SO4 that is ammonium sulfate plus carbon dioxide and water. Now this ammonium sulfate in presence of catalyst like sodium hydroxide, it will be converted to sodium sulfate plus ammonium hydroxide. This ammonium hydroxide uh, will again give you ammonia. Then ammonia plus boric acid that is H3BO3 will give you ammonium plus BO3. Then BO3 plus 3H plus it will again give you H3BO3 that is boric acid. Now we'll see the glasswares. The glasswares which we are using for this particular determination are the digestion tubes, the measuring cylinder, conical flask, burette for titration, and the pipette. So this is the basic equipment for the digestion of your sample, that is Geldal block digestion system. Then fume scrubber, weighing balance, Geldal distillation system, then electrical stirrer and stir bars. Then bottle dispenser capable of dispensing 10 ml of concentrated sulfuric acid. Now what are the reagents which we are using? Those are the first and the main is concentrated sulfuric acid, then catalyst. So here you can see the catalyst which we are using is sodium sulfate and copper sulfate mixture. That how we have to prepare 10 parts of sodium sulfate and 1 part of copper sulfate. Then sodium hydroxide solution 40%, then boric acid 4%, ethanol. Then for titration, standard 0.02 normal hydrochloric acid, which acts as a titrant, and mixed indicator for the determination of end point. Now we will see the procedural flow chart for the same. First step is sample preparation. So in the sample preparation, the sample should be thoroughly ground and mixed prior to the analysis. Whatever your sample may be, food sample or any agricultural sample, whatever, you have to crush the sample, or in fact you have to homogenize the sample properly prior to the analysis. Now next step is digestion. In the particular step of digestion, what happens exactly is the conversion of nitrogen in the sample to ammonia, that we have already seen in the previous slide of chemical reactions. Now prepare 8 numbers of 250 ml digestion tubes for the samples, standard and a blank to be analyzed. After that, the tubes, these are the tubes, nothing but the digestion tubes only, that we are going to fix in the particular digestion block system. So the tubes should be cleaned and rinsed with distilled water in order to eliminate the contamination from previously run samples or foreign material. Now weigh approximately 0.1 to 0.2 gram of sample carefully and transfer the sample into the digestion tube. After this, prepare one standard per batch of sample. That means you have to prepare one standard sample, by adding 0.02 gram of ammonium sulfate CRM, that is certified reference material, to one of the digestion tube. Then prepare one blank for 6 samples to be analyzed. Here, the blank digestion tube will not contain any sample, that is exclusion of sample. Now, add 3 grams of prepared catalyst and 10 ml of concentrated sulfuric acid in each tube. After this, sulfuric acid, here remember, sulfuric acid is to be added slowly and carefully to minimize the boiling or violent reaction. Then repeat the above steps for addition or for all the 8 tubes. That means whatever the catalyst, addition of sulfuric acid, everything what you have added in the sample tubes, the same steps you have to follow for standard as well as blank. Okay. After this, transfer all the samples simultaneously using the tube stand to the digestion block unit. When the sample tubes are in place, place the hood device over the top of the tubes to be digested. Then turn the program on the digestion block such that the set temperature is 420 degrees for 1.5 hours. Then scrubber will turn on and the noxious fumes generated in the digestion process will be neutralized by the particular scrubber system. So clear bluish green color to the digested sample after 120 minutes marks the completion of your digestion. Now remove the digestion tubes from the digester and set aside to cool. If the crystals begin to form as it cools, add 20 to 30 ml of distilled water slowly. Now this is all about the digestion. After digestion, the second step is the distillation. Then in the distillation, separation of ammonia from digested and collection for analysis. Okay, so switch on the distillation unit and allow it to proceed. Set the parameters as NaOH 40 ml, boric acid 25 ml, distilled water 30 ml and time for 10 minutes. Everything you have to set on this particular distillation unit and confirm the distillation unit is connected to tap water and its water flow. Then verify that 40% NaOH supply is attached, steam source is boiling before proceeding for the analysis of sample. Now connect the digestion tube containing the digested sample to the distillation apparatus. The liberated ammonia will distill out into the receiving flask. Now remove the receiving flask from the distillation unit and follow the above process for blank as well as for standard. Now for last step is titration. So in the titration, quantification of ammonia and calculation of initial protein concentration. So put the conical flask containing collected ammonia and boric acid on stirrer, add 1 ml of indicator and gently place the stir bar into the solution. Fill the burette with 0.02 normal HCl and titrate. So you have to titrate the solution against this HCl, 0.02 normal HCl, till the solution turns greenish to pink wine red as color. That means the pink or wine red is the end point of the titration. Now record the burette reading used for the titration. So perform the titration for remaining samples of blank as well as for standard as well. So this was all about the entire process of nitrogen or protein determination. After this, the calculation part is there. So here you can see the calculation formula for nitrogen percent content. That is sample burette reading minus blank burette reading into normality of HCl into 1.4 into 100 divided by weight of sample into percentage recovery. This percentage recovery, how you have to calculate, the formula is also given over here. That is standard burette reading minus blank burette reading. So for the determination of percentage recovery, you will require this standard into normality of HCl into 1.4 into 100 divided by weight of standard into 21.2. From these two formulas, you will get the percentage nitrogen content. But you have to convert this percentage nitrogen content into protein because you are finally estimating the protein content. So protein percent is equal to nitrogen percent into 6.25, where 6.25 is the protein nitrogen conversion factor. So this was all about the protein content from food as well as any agricultural product samples. Thank you for watching our video. Please like, share and subscribe to our YouTube channel for more such informative videos. Thank you.